afternoon, and I want to thank you all for coming today to Cancer Center Grand Rounds. It's a real privilege to introduce today's speaker because I think we all recognize that a central tenet of our cancer center and our cancer hospital is moving great science into the clinic. And I can think of few individuals who have better championed that fundamental principle than our speaker today, Jay Bradner. Jay is somebody I've known for many years from both our times at Harvard. Jay is a graduate of Harvard College and then subsequently, I guess, medical school at the University of Chicago. He did his residency at Brigham and Women's Hospital, his medical oncology fellowship at the Dana-Farber Cancer Institute, and then did an extremely productive postgraduate training in the laboratory in chemical biology, ultimately emerging, leading his own laboratory, which really has transformed, I think, the landscape of cancer therapies in terms of innovations and starting new drugs, new products. About 3 years ago, Jay was recruited to his current position as president of the Novartis Institutes for Biomedical Research, where he has moved that talent in science, in organization, in vision to move things into the clinic at a much grander scale globally and across disciplines. So it's a real pleasure to introduce Jay, and thank you for coming.
Can you hear me okay? Can you hear me okay? Yeah. All right. Charlie, thank you. And it's a great honor and privilege to get out of the shop for a little bit and to come back to a top academic center and talk about our work. One of the joys of working in such a well-resourced, and I mean that intellectually, a biomedical research environment is this sort of sense of wonder and ambition and bravery in science. But one of the downsides is being disconnected with people that you've loved working so closely together. Charlie was my GI attending when I was a medical oncology fellow. I disappoint you by going into blood cancers, I'm sure, but also the very first person I called when my dad got sick with pancreatic cancer. It's wonderful to see you here leading. I brought a slide deck that has just a little bit of chemistry. And after a big belly full of sandwich, this can be very sleep-inducing. And I recognize that. It's fine to sleep, but snoring is rude to your neighbor. I won't mind one bit. But I wanted to share something with you that is exciting to me, both scientifically and medically. And it's the way that my own thinking around what's possible in drug discovery has really changed, moving from a top-flight academic center in the biotech environment to NIBR, which was not an institution I knew a lot about. And so hopefully I'll have a chance to reintroduce the Novartis Institutes to you through this lecture. And specifically, that sense is no longer this idea that something is druggable or not druggable. I've seen just enough examples in my time at Novartis of what I might have regarded as intractable protein targets taken down by coordinated efforts in ligand discovery and deploying new technology platforms to now believe that nothing is undruggable. And so I would say we're just at the beginning of the end of undruggable. Yeah, I have these disclosures to share with you. Hopefully they've all been captured by the CME group. And as you know, I'm an employee of Novartis, which is the intergalactic Swiss pharmaceutical company that owns the Novartis Institutes for Biomedical Research. So this is not to dumb this down for all of you. This is the way that I, as a medical doctor, still understand druggability. That like shells, pasta, my daughter would put this on her pinky, and that's sort of like the drug fitting into the hydrophobic pocket with excellent molecular recognition, shape complementarity. When there's a pocket in a protein, medicinal chemists today, pretty good at sculpting a molecule to pair up the partial negative and partial positive charges, to extrude water where it's high energy, ultimately leading to a stable association to activate or to inhibit a protein target. This is MedChem 101. And I think this is well exemplified by our prior work on bromodomain inhibition, kinase inhibition, and. There's emerging protein space proteins that maybe could be druggable, but it's not yet been demonstrated. Druggable is a term of trade in science that means that there's already known to be a molecule that binds it, a small molecule that binds it. Could be a natural molecule like a sex steroid binding the estrogen receptor, or it could be a drug molecule. And so part of our charge in industry, as in academic chemical biology, is just to put more proteins in the win column. And so this, I think this is called rotelle. We call them wagon wheels in our house. This looks a lot like the WD repeat domain. And this protein fold had never had a ligand for it before, but it does have a little pocket in the middle, like the hole in the donut. And scientists at our Shanghai site called CNIBR made a first small molecule to bind and inhibit the EED WD repeat domain, which is a regulatory component of the polyclonal repressive complex. It's the off switch for many genes. And so this molecule turns on healthy genes, now open in B-cell non-Hodgkin's lymphoma. What was cool about this project is there's actually hundreds of these WD repeat domains. And so like finding the first kinase inhibitor in Gleevec, this really opens up the landscape of new target space. But what happens if your target is like spaghetti? It has no pocket. In fact, when you engage with it, it changes shape. And that you can't get a high affinity interaction with spaghetti. Then what do? We actually lack technologies for this, because all of medicinal chemistry is predicated upon a stable interaction, a key fitting into a keyhole. What if there's no keyhole? And such is the challenge with the group of proteins that I cut my teeth on as a scientist called transcription factors. These are the proteins that bind DNA in a sequence-specific way, and they turn genes on or turn genes off to make a cell type of interest, or for that cell type to change its cellular state, pre-inflammatory to post-inflammatory, pre-metastatic to post-metastatic behaviors. This is MYC bound to MAX, sitting on its EBOX DNA, and MYC is in blue. And you can't find a pocket on this complex. And in fact, when MYC releases MAX, it becomes spaghetti. And so it's like setting up a chessboard. You wouldn't have to do this because I'm very bad at chess, but perhaps to play my son, you would initially set up the board, and then you would start to execute your strategy. And so when I got to NIBR, we thought that to take down these undruggable proteins, we would build an organization called CBT, Chemical Biology and Therapeutics, 400 scientists on 2 different campuses, and their charge is to reinvent the science of therapeutics. And sometimes there's a target in mind like MYC or another gene control factor, but most of the time we just expect therapeutics to sort of emanate from these efforts. It's my big bet, and if I remain employed, you will know that it's working. And so this is an excellent example of molecular recognition from one of our MedChem programs. This is a small molecule binding into the active site of a protein. And you can see that the way that the reds and the blues line up and the water molecules are all coordinated, and the bend on this molecule emulates the pore of this protein. When we have pockets, we can execute. But when we don't, we have a challenge. If you're interested to learn more about the science of druggability and not just this simple parlance, there's a whole field around it. We're using 3-dimensional crystallographic data. Computers can survey to, almost like rock climbers, find the crevasses within proteins and assess their suitability for drug discovery. And we deploy these technologies all the time. Our preference is to make keys for keyholes. But where no keyhole exists, we're stuck. And this really came through loud and clear in cancer medicine when sequencing efforts started to reveal the pathobiology of these diseases. And as a blood cancers doctor, a great many of the somatically altered gene products that drive blood cancers are not kinases. There are these gene control factors like MYC and others, like Notch, that are not overtly druggable. So we have a great need in science right now to proffer more targets, context-specific, synthetic lethal. But there are a lot of targets that we've known about for 30 years and we have no drug. And that starts to feel unacceptable. And so I thought, for my time at NIBR, where we have, for once, the resource appropriate to action brave biomedical research, that we were going to put all of our eggs in that basket and reach for the highest hanging fruit, leaving the lagging strand, the very fast lagging strand of science to the biotech industry, who is also very innovative, but is often under extreme time pressure which thankfully, at a company that's over 100 years old, we are not. The second time that it was driven home that these undruggable targets are high-value targets was the outcome of a massive study that Bill Sellers did when he led Novartis Oncology, now led by Jeff Engelman, to map all the Achilles heels of all the cancer cell lines we could find. Hundreds of cancer cell lines, hairpin and ultimately CRISPR screens with thousands of gene products, looking for the specific dependencies of each specific cancer type. And all the caveats of two-dimensional petri dishes and plastic cell lines immortalized forever. But there's a lot of learning there. And what we found, regrettably, is that in the kinase family, all the targets that we found already had drugs. But the blue, the open water of drug discovery, mapped to these transcription factors and other undruggable targets. And so there wasn't that much low-hanging fruit left to collect. What we look for are these outliers where the estrogen receptor is very important for a subset of cancers, or the MITF transcription factor, which is a stage-specific transcription factor required to make a melanocyte, is required in melanoma. And the learning there is fantastic, but the actionability for drug discovery is little. All the more reason to get to this high-hanging fruit to try to reimagine this science of therapeutics. If we're going to make a KRAS inhibitor, it's going to take outside-the-box thinking, like Jonathan Ostrom, thinking that he could bind into the G12C cysteine on KRAS. Well, what do we do for G12D? We have to figure that out. And so I want to introduce a term that I'm falling in love with that defines a class of molecules that we and others, I should say, and others in this room, and I'll be sure to call them out, are innovating, called molecular glues. Whoa. And I'll work to define what a molecular glue is. Don't feel any obligation to use this word. I'm not even sure it belongs in scholarship. But in the parlance of drug discovery at Novartis, we're in love with these molecular glues. All right. So some molecular glues work by like a full Nelson, like a wrestling move that like locks proteins in an inactive conformation. And because they work within one protein, we call them intramolecular glues. And a great example of this is a molecule I'd love to get you excited about because it's now in human clinical investigation. And Dr. Herbst, I'd love your feedback on this. It's been a long-standing challenge to make a drug for protein phosphatases. Kinases, after Gleevec, easy. Compete, ADP, inhibit the kinase. Phosphatase is a little harder. It turns out phosphatases have a pocket. It's where the phosphate binds. It's easy to drug. High-affinity probes have been made for many phosphatases. The challenge is those probes are negatively charged like phosphate, and they don't get into cells. And even with all the pro-drugging sophistication, nobody has made a phosphatase inhibitor until now. So SHP2 appealed to us as a great target because it's a protein tyrosine phosphatase that opposes a lot of oncogenic signaling off of receptor tyrosine kinases. And we used experiments of nature, such as Noonan syndrome, that reveal activating mutations in SHP2 to understand residues across different of the 3 soluble domains of SHP2 and how they might work together. In Leopard syndrome, there are inactivating mutations of SHP2, and they also reveal ways that you might take down the SHP2 phosphatase. SHP2 is required for receptor tyrosine kinase signaling in cancer. There's a great literature and great reviews on this. It's also been recently identified as to be an important downstream transducer of PD-1, as we just spoke about. And so there's a chance that if one could make a SHP2 inhibitor, it might have checkpoint inhibitor-like properties. And when we did our big hairpin screening and CRISPR screening experiment, there was asymmetry in the data such that a small number of RTK-dependent cell lines were enriched for SHP2 dependencies. So all of these things add up a very compelling case to try to make a SHP2 inhibitor. And so the innovation that the NIBR scientists did is they recognized, having done other phosphatase screening work, that they were probably going to find active site inhibitors. And so we screened 3 million molecules with the full-length protein, and then we threw everything out that worked on the catalytic domain, which was almost everything. And it left us with a very small number of starting points. And in that way, in the open and active conformation, molecules that would bind the free active site, which could be great phosphatase inhibitors, get thrown out. And we looked then for molecules that might enrich for an inactive conformation, this or otherwise. In parallel to this 5-year drug discovery activity, much of the scientific literature was well validated in-house, establishing SHP2 as a bona fide dependency in solid tumors characterized by active MAP kinase signaling from receptor tyrosine kinases. But still, we had to be very sensitive to the historical experience here. Over the last almost 20 years, there are probably 35 kinase inhibitors that are now active in human clinical investigation, and there are 0 phosphatases, and that's not for lack of trying. The phosphatase program at Novartis was retired formally after 10 years and probably 100 associate years worth of work. And so to reopen this, we had to be confident that we were doing something very different. So as I shared with you, we conducted high-throughput screening with a signal-on type of assay, which tends to produce fewer false positives than a single-off type of assay. We did exhaustive biochemical characterization of what SHP2 would look like with and without its activating peptide. And then we threw out all the active site inhibitors that worked only on the PTP domain, keeping molecules such as this one that I'll tell you more about that seem to work only in the presence of the full-length SHP2. One of the sub-libraries studied was an index kind of test run screen of 100,000 molecules that ultimately gave us our lead compound, which was this aminopyrimidine that hit in the first screen. It's low potency at about 12 micromolar in a cell-free biochemical assay, but it got us excited because this molecule, as well as this one here, had this kind of common chemical motif. And though we couldn't, advance this molecule because of toxicity on sodium channels, seeing the enrichment of a potentially common, what we call pharmacophore, inspired us to put some medicinal chemists on the case. And by some, I mean like 45. Now, we also had a strong crystallography effort going on in parallel, and this turned out to be essential. There was no full-length SHP2 crystal structure, and we were working with molecules that would work only in the context of full-length SHP2, and so our crystallography group heroically obtained a high-resolution crystal structure of SHP2 in the presence of one of these prototypical molecules, and lo and behold, discovered that it was in an inactive conformation. It was glued. Now, if there are chemists like Craig and others in the audience, hopefully this is exciting for you. In other audiences, this is like you flex your muscles moment. We took this very low potency molecule and then iteratively, using the guidance of structural biology, built new bridges, such as this with freeamine over to a side chain carbonyl. We worked through all of the challenges of hERG activity and off-target channel activity, ultimately taking a 12 micromolar molecule to 70 nanomolar in cells without the molecule increasing its molecular mass. Usually to make a molecule potent, it gets much, much bigger, like all the ones I made when I was a professor. But here with these first world medicinal chemists, they improved what's called the ligand efficiency, the potency of binding relative to the size of the molecule. It's an extraordinary achievement. And that turned out to be important because with 3 domains, east, west, and the middle, and the Midwest, on this molecule, it really couldn't get any bigger if we wanted it to remain orally bioavailable. And so that's the way the molecule works. It starts with an inactive conformation, and then an active conformation and locks it in inactive. And you can see the pocket where phosphate binds is completely unaffected, although there's a loop that forms when we close the clamshell that ultimately extrudes substrates. Here's the molecule, and you can see I've color-coded each of the 3 lobes, and the molecule actually bridges all 3 lobes. It glues 3 things together. Yeah, it's amazing. And as this molecule was optimized, we naturally wanted to know, would it also work on SHP1, which would present, we would probably still develop it, but there would be toxicity associated with SHP1 and no benefit. And interestingly, when we crystallized SHP1, what we found is that the channel exists between these 3 domains of SHP1, but it's just big. And because it's big, it's flooded with water, and the molecule just gets washed right out. And so I think because of its allosteric binding site and the complexity of its 3-dimensional molecular recognition event, this molecule is very, very selective for SHP1. And in our panel of more than 70 phosphatases, and by phosphoproteomic analysis, as best we could tell, it only inhibits SHP2. So it's an excellent probe. And I put this up just as a reminder to say, in our own Swiss way, we did an almost irresponsible amount of biophysical characterization of the molecule and ultimately found that it binds with 1-to-1 stoichiometry. It has excellent kinetics with a slow off rate commensurate with locking a protein into a glue-like conformation, and advance the molecule into preclinical studies. The molecule that ultimately we brought to patients has a very long half-life, which we thought was an asset to maintain inhibition of tonic receptor tyrosine kinase signaling, which has been characterized in lung cancers and in liquid tumors is very, very important. So with an excellent and durable plasma concentration, phospho-ERK signaling downstream of SHP2 is markedly suppressed. Like the shRNA knockdown, SHP099, which is our chemical tool, shows excellent suppression of tumor growth, and we did observe responses in a panel of primary human xenograft models that Bill Sellers and colleagues collected over many years. So that's the idea of how you can approach a historically undruggable target, and by only interacting with that protein in particular, lock it in an inhibitory stranglehold. And so we're starting to wonder, could we do that with MYC or one of these other spaghettis, like the way my daughters would like tie a spaghetti like in a knot or something like that. That's not working yet. Maybe I'll come back sometime. The second type of glue is where you take one protein and you glue it to another protein. There's a bunch of examples of this in nature. Surely you know of the medicine called RAD001 from Novartis. It's an mTOR modulator. And rapamycin, the natural product, actually locks a phosphatase and a regulatory protein into a chemically induced dimerization event. And so inspired by nature, many scientists, most notably among them Craig Cruz here on your faculty, have worked to try to lock proteins to other proteins. As Stu Schreiber, our mentor, had famously exemplified with chemical dimerizers and engineered systems. We worked to glue proteins to the trash system, to the ubiquitin proteasome pathway, with the idea that sometimes when we drug something like the bromodomains that I used to study, we drug the wrong part of the protein. It's like if you wanted to shut me up right now and you put like duct tape on my knee, it would not work. And that's what we sometimes do in drug discovery. We make a drug for, I don't know, CBP, the histone acetyltransferase, but the part we drug is like the bromodomain. And then the acetyltransferase just goes on ticking. Sometimes the part of the protein that you want to drug, the active site, is just not druggable, like KRAS. You can't get GDP out, but maybe you could stick to the other side. So we saw this as an opportunity to hit some previously otherwise inaccessible targets. But there's another advantage of degrading a protein, and that is if you make the whole protein go away, then all of its functionality is gone, meaning the impact, the efficacy, the Emax of such a molecule is even better. And then secondarily is that these molecules could behave almost like little molecular catalysts. Like if the protein were degraded, but the molecule wasn't, at a very low concentration, you could whip through a lot of protein, which would make these extraordinary medicines potentially. And the idea came to me when I was reading Hiroshi Honda's science paper in 2010, where Hiroshi was trying to understand how thalidomide works. And so he went fishing into the human proteome. And he pulled out a ubiquitin ligase called cereblon, which is an E3 ligase, the last catalytic step that puts the trash sticker ubiquitin on proteins of interest. And I got excited because if Hiroshi could pull the garbage man down onto a bead, maybe I could trick the garbage man and move it around the cell, destroying proteins of interest. In this regard, there are 3 enzymatic steps, E1, E2, E3. Hopefully with Craig on campus, you know this chapter and verse. And that these ubiquitins are ultimately polyubiquitinated and transferred onto the biological substrate. And we thought, could we trick this with a small molecule, imperceptibly small, to this big molecular machine to bring in an aphysiologic substrate and destroy it? And like a lot of our best ideas, this has already been invented. In 1999, 2 scientists in industry had this idea that could you take a protein and wedge it to the ubiquitin proteasome system? But they never got it working. Craig did. In early work from your lab, Craig, working with Ray Deshays with these long polypeptides, appending a targeting agent at concentrations of 20 to 50 micromolar, there's evident degradation of the target protein, and the field got very excited about the possibility that this could really work. But there were challenges at the time we got started on this in my academic laboratory. The first is that these by and large worked with peptides, which don't get into cells very effectively, or you had to engineer approaches like put a targeting motif onto your protein, which you can't do in cancer. It's just sort of born that way. Or that the molecules couldn't work in vivo, perhaps because of low potency and lack of drug-like properties and a lot of important biologies in vivo, especially in our patients. And so we undertook to try to create an all-chemical solution to these challenges. This is published, so I just brought the one slide. So we took a derivative of thalidomide and we attached it to the bromodomain inhibitor from my lab called JQ1 with a chemist who trained with Craig here at Yale called Dennis Buckley, who came up to Boston to do this work. It didn't work right out of the box, but by doing linkerology on this spacer between the two, we could kind of figure out the distance required to engage both the target and the garbage man. And so this molecule, DBET1, we characterize biochemically. It has the characteristic hook effect, where at high concentrations, you compete the molecule off by a second molecule. But at just the right concentrations, like Goldilocks, then these two can lock themselves together. And when they do, we saw sub-nanomolar, in some cases, degradation of target proteins in cells. And this was very exciting. We sent the cells off. These are leukemia cells for proteomics. And we were very excited to learn that only the proteins that are targeted by the targeting warhead are degraded. And so this really is sort of co-opting the highly evolutionarily conserved mechanism of protein degradation. And this was very active in leukemia clinical trials in mice and is now being developed by a company called C4 Therapeutics that I have no connection to anymore now that I am at Novartis. And so after this manuscript was submitted, we got feedback from the reviewers and they said, this is like amazing, do it again, which is kind of harsh. Like, you know, when you've been working on something for 5 years, like, do it again. And so, but it was a fair point. Like, if this was to work, it should not just work just once. You should be able to do this again and again. And in the bromodomain family, beyond JQ1, the first bromodomain inhibitor from our group targeting BRD4, scientists at GSK, at Oxford University, and others have made, MD Anderson, a number of bromodomain binding molecules. And many of them bound the bromodomain, but they didn't do anything. The TRIM24 molecule bound the bromodomain of TRIM24, but it's really an E3 ligase for P53. And so we like drugged the wrong thing. Duct tape on the knee. CBP had a bromodomain, and it does something, but not as much as getting rid of the whole protein would. And so systematically, we started to append these bromodomain ligands onto the phthalimide. And I will say, with a little bit of luck and a lot of chemistry, all of these have worked. And it's wonderful now, being out of academia, to follow this literature and see just an explosion of science from many, many different labs using phthalimide conjugation chemistry to degrade new targets. Here's a paper we just wrote on BRD9 with a BRD9 bromodomain targeting ligand. Mass spec, only BRD9 is degraded, and we could validate this as a potential target in leukemia and sarcoma. Here's the inactive TRIM24 ligand. We appended a molecule from Craig and Alessio Ciulli's lab, and this targets the VHL E3 ligase, and there's TRIM24 is degraded, and leukemia cells would arrest. And then here's a very promiscuous molecule that targets CDKs, many different cyclin-dependent kinases, many different protein kinases. But we were surprised to learn when you start with a very non-specific targeting agent, sometimes you can get a very selective degrader out the other end. And this is because what we were able to show in their very first study is that these molecules work like molecular catalysts. If you take two cell lines, one has the ubiquitin ligase and one doesn't, our BRD4 degrader will kill them both. But it will kill the cells that have the ligase at 1,000-fold or more better concentration. So the molecule gets in, and it disrupts all the BRD4 at stoichiometric concentrations. But when it's whipping through and degrading everything, it can work at very, very low concentrations. And we think that has a lot to do, and also the way that ubiquitin is transferred, with why non-selective molecules, sometimes retired by pharmaceutical companies for toxicity, can be like resuscitated as very selective degraders. In our work and in the literature, we now see actually more than 100 proteins have been degraded using this and similar technologies, and the field is wide open. Here in Connecticut at Arvinas, a molecule very similar to the BRD4 degrader was thereafter shown to degrade BRD4, which is cool. And Alessio Ciulli at Dundee in England, who's an outstanding scientist who also trained here at Yale with Craig, took our JQ1 molecule and put on the von Hippel-Lindau. Now this is a different E3 ligase, and he degraded BRD4. So it's like open season now on these proteins. What's exciting is also to watch how quickly these are advancing into human clinical investigation. There's probably 8 dedicated biopharmaceutical companies studying this, and almost every major pharma has a collaboration in this area. And so we expect very soon for this science to reach human clinical investigation. So I think there could be new drugs that come from this. I mean, there are challenges. These are big monsters. And getting these orally bioavailable with good PK into the brain or not into the brain, these remain obstacles to the development of medicines from this. But we and others have solved many of these challenges on a case-by-case basis. But it is heroic chemistry. How can this matter for primary scientific investigation? Well, what if your protein of interest that you study doesn't yet have a ligand? Like, what if there's no binder for your protein and you want to get in on this, or you want to see what life would be like degrading this, but there's no starting point? Then what? What if you study like really fast biology? Like I study transcriptional biology, and you could do like a hairpin screen or a hairpin experiment for MYC and then wait 48 hours and see what MYC does. But by then, you're seeing the secondary, tertiary, quaternary effects of MYC, not the role of MYC. Metabolism is fast, transcription is fast, translation is fast, synaptogenesis is fast. These would be great probes to study this biology, but what if the probe doesn't exist? And so we created a solution for this we call DTAG. It's recently published in a pair of papers in Nature Chemical Biology. Here, we took a protein and a molecule from Ariad Pharmaceuticals. It's a 12 kilodalton tiny little protein called FKBP12, and it has a hole in it. And then we made a ligand, a novel ligand in our case, that binds into that hole. And so now when you hook that up to the degrader, only that mutant is degraded. Even the wild type FKBP12 is not degraded. And with CRISPR, you can drop ship this into the human genome anywhere you like here, next to BRD4, my favorite protein. And then you can see the way wild type FKBP12 is not degraded, but the conjugated protein, which we left a hemagglutinin tag on there for Western blotting, is degraded. And this has proven really handy. We used it to validate a new cancer target called ENL, which has what's called a Yates domain. And before we made a Yates inhibitor, we wanted to at least ask the question, like, is Yates containing protein ENL a good target? And so lacking a drug to make a degrader, we put in the DTAG, and there it is selectively being degraded. I'm sorry, this is a hit from a CRISPR screen. There it is selectively being degraded, and we could demonstrate that it arrests proliferation. And because this works so fast, we can degrade ENL and ask 15 minutes, 1 hour, and 8 hours later, what does ENL do? And it turns out that ENL is very important for the maintenance of what's called the core regulatory circuitry of a cell. It's part of cellular identity and transcriptional elongation globally. And I mention this because this DTAG system could be very useful for you in what you study, and it's all freely available on Addgene, and hundreds of labs are now working with this system, including our labs at Novartis. There's no reason to show this except I just think it's kind of cool. This is the DTAG on GFP, but not on RFP. And so you can watch cellular processes and take just one component out of the system and then see how your cellular biology of interest responds. We made a bunch of other kinds of glues, and I'll just give you some snapshots as opposed to telling the full story, but I think each of them are cool stories unto themselves. You must know about the disease called spinal muscular atrophy. It's one of the most common genetic causes of death in newborns and children. It's a disease where children are born without a copy of the SMN1 gene, and then depending on how many extra copies of SMN2 they have around, they either have debilitating neurodegenerative disease, because they only have 2 copies, or they have a delayed neurodegenerative disease, because they have 4 or 6 copies. And it's a horrible disease. And so at NIBR, you can't make a protein like reappear. That turns out to be really hard. And so what we did is we learned from experiments of nature that there are often splice junction polymorphisms of SMN2 that change how much protein is elaborated. And we asked the question, could we, in our 3 million molecule collection that we had 5-6 years ago, find a molecule that would fix the splicing so that each transcript just makes more protein, a splice site modulator. And there's no example of this, but this is the science of therapeutics. This is published now in Nature Chemical Biology, and this is a model of how the molecule works. The molecule turns out to bind RNA. It binds RNA in a sequence-specific manner and in a context-specific manner, 3-dimensionally, when it's loaded onto the SNRP, onto the spliceosome. So if you can drug RNA, maybe that's a way to drug something where you couldn't drug the protein itself. And we're applying this learning now to neurodegenerative disease targets, MYC, and other targets as well. This molecule is now in human clinical investigation, and this horrific disease, as the NeuroNext population study showed, this is a performance score called CHOP-INTEND, which is the regulatory endpoint. And over time, by 2 years of age, these children are often on ventilatory support or no longer alive due to severe respiratory illness. And you could see for the very first time, this medicine is actually increasing the CHOP-INTEND score. More on SMA in a minute. The wildest kind of glue is a glue that a friend of mine, Asim Ansari, just reported a year or so ago. Compartmental glue. Whoa. So now we have glues that work on single proteins, glues that work on 2 proteins, glues that work on RNAs and proteins. Like, what if we could move a protein to a different part of the cell? Glue it someplace else. That would be really interesting. Put KRAS somewhere you'd rather not have it be, or move a transcription factor around the genome. Well, there's a disease called Friedreich's ataxia, where a gene called frataxin is not expressed because the promoter that drives that gene is silenced. It's epigenetically turned off. Perfectly good gene, sitting right there. But these GAA repeats in the promoter cause the switch to never turn on. And so Asim and I wondered, could we take a little bit of his chocolate and our peanut butter and make a medicine for this disease? Asim's lab studies triple helix formation, and with these long polyamide molecules, you see these amide bonds, polyamide molecules, some of them are sequence specific, and this one in particular binds GAA. And so we hooked up this GAA binding molecule to our bromodomain inhibitor, but not to use it as an inhibitor. Here, we want to recruit the genetic activation machinery over to the silent GAA domain. And quite unbelievably, the frataxin gene turns on, shown at the RNA and protein level. It's highly selected for just the frataxin gene. And we got excited, and I got a little bit anxious, because you often hear how academic science doesn't repeat in industry. And I had just gone from academia to industry. I don't want to be both sides of that equation. And so we took this molecule into the labs, and we tested it in the frataxin cells from patients that we turned into iPS cells and then back differentiated them down as myocytes and neurons. And what we found is in these primary isolates, differentiated along multiple lineages, that indeed the frataxin mRNA is able to be activated, making protein 2 and reversing some of the metabolic phenotypes with this small molecule. We've glued a transcriptional elongation factor to a repressive part of the genome, like an artificial transcription factor. I think it's really cool. These stories were just to sort of exemplify this concept of what our chemical biology organization does, which is to imagine molecules with new types of behaviors. For today's lecture, these are the molecules that have multi-dimensional behaviors, the way that proteins have multiple domains and multiple dimensions. Other technologies that our group is interested in is to turn the 3 million molecules of Novartis into 3 billion molecules. And we're already at 1.7 billion over the last 3 years by building molecules, not in solution in Erlenmeyer flasks, but on DNA. I don't know if you followed this chemistry, originally out of academia, but now perfected in Switzerland. We take a barcode with a single molecule on it. We add a second molecule with a chemical conjugation, and that gets a second barcode. Then we add a third molecule, and that gets a third barcode. And if you permute 1,000 of this, 1,000 of that, and 200 of those, the numbers start to add up, and you make massively large chemical libraries into which we can dip a protein of interest, pull it out, and sequence the DNA that comes with it. This science has been around for a while. There aren't as yet any DNA-encoded library drugs, but we're hopeful that in 2019 we'll have one of the first. We're crazy about digital. And I think that there's a lot of hype out there about the contribution of artificial intelligence and machine learning to science and medicine and drug discovery. I can say that at NIBR, we don't think that it's going to kind of like solve everything, but we do believe it's like an augmented reality, that the chemist doing molecular dynamical simulations with machine learning will put out of work the chemist without it. But we don't think the robots are coming for us all. I hope to retire just before that happens. I didn't talk about it at all today, but another big investment we've made over the last few years is in advanced cell and gene therapies. We're crazy about the idea of CRISPR in a couple of contexts, vandalizing the human genome in such a selective way as to eliminate the negative regulatory region that drives fetal hemoglobin, turning it back on to potentially provide curative cellular therapy for sickle cell disease. And we're very interested in gene therapy as well. Spinal muscular atrophy might benefit with a compensatory increase in SMN2, but the right way to approach that disease is to give these children back the gene they were born without. And so last year we acquired a company called Avexis in Bannockburn, Illinois. It's a Chicagoan, I just love that. We used to go to Bannockburn to buy milk. And this is a remarkable company out of Nationwide Children's, led by Brian Kaspar, that uses adeno-associated virus 9 systemically delivered to get into the brain and to the spinal cord and deliver the SMN1 gene. And it's very much maturing in real time, these data. But if you can see, the probability of survival now out to 78 weeks and longer of these children who've received the gene therapy is quite striking. And it's quite a powerful testament to the promise from so many years ago now being realized in gene therapy. So, just to be provocative, I don't think there's anything we can't drug, but these projects take a lot of time and a lot of creativity. And our very best science at NIBR is done not in isolation. And so the reason they let me out to come talk to you is for this slide, is that our company, like many others, has been very inwardly focused. We have maybe one of the largest, if not the largest, research infrastructure in the biopharmaceutical industry. And to build that took a lot of internal focus. But now we seek to make it more accessible to innovators just like you. It could be your biotech company, or ideally it would be you. When you walk through the labs at NIBR, it's like a visceral feeling of how serious science is on that scale. And the most wonderful thing about it is there's no intermediate strategy. We publish a lot, 730 papers last year. There's no barrier to you publishing. But our favorite projects are ones that will take 10 years ultimately to do, because those are the ones that may not get done by peers that lack a committed research investment. By the numbers, it's 6,000 scientists on 6 campuses. We have 340 research projects in 8 disease areas. 40% of our investment is in cancer. And with a research budget of about $9 billion, like nothing's off the table. It's all about who has the best idea. Sometimes at NIBR, sometimes it's at UPenn, where we did all of our foundational CAR T-cell work so many years ago. These are the other disease areas where we work. And if you work in any of these areas, like neglected and tropical diseases, for example, and I know there's great science on this campus in neuroscience, please do connect to us, challenge us with your ideas. I knew that there would be a lot of people here who are clinical investigators like you, Pat, and colleagues. And so this is the whole portfolio and all the people. We've recruited Jeff Engelman from Mass General to lead oncology. Lily Petrozelli leads all of phase 1 clinical trials. Peter Hammerman does all of our on-treatment biopsy mechanistic work, and Glenn Dranoff leads immuno-oncology. You can email me or connect directly to them, jeffrey.engelman@novartis.com, and help us put these molecules to work. Our very best medicines that are prescribed and used around the world are almost never prescribed for the purpose they were originally invented. It happens, like gene therapy for spinal muscular atrophy. But Gilenya, our multiple sclerosis drug, was supposed to be a solid organ transplant drug. And Cosentyx, the psoriatic arthritis and ankylosing spondylitis drug, was supposed to be a rheumatoid arthritis drug. And what we've learned is over time is by providing these molecules to the scientific community to put the work and figure out how best to use them within the window that we can support them, the period of exclusivity, is our best chance. It's about 18 targeted therapies and a number of immuno-oncology medicines. And we're desperate to find uses for these alone or in combination with our drugs or other companies. To us, it just doesn't matter. I'll leave you with a couple of recent achievements of our clinical development that we're proud of. The first is we recently had the very first phase 3 clinical trial ever to report positive for ADP competitive PI3 kinase inhibition in breast cancer. And just like our proof of concept study suggested, which was performed in a very novel way in only PIK3CA mutant patients, that in the phase 3 clinical trial, on top of fulvestrant in ER-positive patients, there was a doubling of progression-free survival. And remarkably, in a disease that is well studied and ambitiously pursued as breast cancer, this would be the first oncogenic mutant-directed or targeted therapy for breast cancer. which we are hopeful. It's being well received by the community. We await the regulatory consideration. We're hopeful that this could be an important medicine. We also were the first to develop CAR T-cell therapy with UPenn. We tend to study the B-cell malignancies because we have to focus. And so CD19 CAR T-cell therapy called Kymriah is now approved for pediatric ALL and diffuse large B-cell lymphoma in adult and children. And there's a stable of next generation technologies coming that will hopefully make it easier to administer, to manufacture, and expand the use even beyond B-cell malignancies. And then finally, the most provocative thing that we've done, and I know Yale is working with us on this, so I wanted to thank Dr. Herbst for working with us here today, is that it's like the most interesting thing we found that nobody's working on. I just don't understand it. As a cancer doctor, I will say, I am very humbled by the challenge of making definitive medicine for advanced solid tumors. Cancer is very fast at evasive resistance. It's very good at it, and it's inborn in the genotype of the tumor once it's radiographically exemplified. And so I become a believer that we need to prevent cancer from happening, but with like mechanisms pursued with prevention in mind, not drugs that work in late stage, advanced to early stage, then considered like tamoxifen for prevention. Like, what would you make if prevention was what you were trying to do? And we think immunomodulation is rich with such opportunities. We did a massive clinical trial of a medicine that inhibits interleukin-1 beta called canakinumab, or it's marketed as Alaris, for pediatric febrile syndromes and other uses. And we studied the inflammatory hypothesis of atherosclerotic heart disease. As you perhaps have heard Paul Ridker speak on many occasions. Is atherosclerosis driven by macrophage inflammation? And we had the right molecule for this. And so we did a huge clinical trial, and indeed, compared to placebo, we prevented the time and dose-dependent effect of inflammation on sequelae after a first myocardial infarction. And the effect was modest, and ultimately we got a complete response letter, and it will not be FDA approved for this indication. But the study was so big in people of a certain age, and as being in that age, I can say that we could ask other questions like, did you get cancer? And this was all built into the study prospectively because interleukin beta was known by our scientists at Novartis to play a role in the metastatic spread of cancer, the formation of the metastatic niche. And in a dose and time dependent way, canakinumab reduced the incidence and death from lung cancer. Now there's a very short window until this drug goes off patent, and we're very motivated to try to find partners to study this as cancer prevention therapy, and as we're studying it here at Yale, adjuvant therapy. Thanks for letting me share some of the stories from our science, our labs, and I take any questions. And again, Charlie, thanks for the invitation down.